All right, next is Griffin Weber, who's going to talk about the Mass Data Consortium, and he's associate professor at Harvard Medical School. So Griffin, and also, of course, the lead of all the public health stuff that we talked about earlier today. Griffin organized all that. So, Griffin.
All right, thank you. I'm going to talk about the Massive PR Federated Data Network. I actually don't think I can present this better than Dean Daly did earlier this morning, so I will try to I'll try to do as best as I can, but he did a great job of describing this. A couple acknowledgments, disclosures. A lot of this was supported by Harvard Catalyst and Massive PR and many others that contributed to this work. So the Massachusetts Consortium for Pathogen Readiness, it was created in 2020 to address the COVID-19 pandemic and it expanded to other pathogens with potential, with pandemic potential. It has a number of different partners, both Harvard affiliated hospitals, as well as other hospitals and research centers in across Massachusetts and public health agencies that are collaborating with us. The dean over there is the executive director. She can tell you a lot more about Mass CPR. One of the main activities it does is awards pilot grants to now over 125 different research projects with over 1,000 publications that have come out of this. Two of the main programs are its biospecimen program and the data network that I'm going to talk about today. This data network has been in planning since 2020. It takes a long time to build the trust between institutions and to figure out how the data sharing will work. This is a high-level picture of the model. On the left-hand side are individual hospitals that store their data in these I2B2 systems. We started with Beth Israel Lahey, MGB, and Dana-Farber data. We're expanding it out to additional healthcare institutions. We're pulling in first wastewater data. We heard about that earlier today, as well as potentially others in the future from the public health data sources. And we're exploring how to integrate the biospecimen data that Mass CPR has organized, as well as internal biobanks within the various hospitals. These data remain behind the firewalls and locally within each institution. And then we have a federated network system that takes users' queries, distributes them out to the sites. They get computed locally, and then just the obfuscated aggregate results that get combined and returned to the users for various applications and use cases, such as retrospective infectious disease research, clinical trial preparation, pharmacovigilance studies, look at side effects potentially of vaccines or treatments. Eventually, access to the biospecimens and resource allocation. We can make multiple copies of data, but biospecimens are limited resources, so you want to make sure that those are allocated and aligned with the correct research applications. And then the overall use of this network is for improving pathogen readiness. The network is live and available to investigators at Beth Israel Lahey and MGB for research in public health use cases related to infectious disease. The underlying programs behind these are I2B2 and Shrine. You'll learn a lot more of them later today and particularly tomorrow at this symposium. Informatics is informatics for integrating biology bedside, I2B2. It's an open source tool that integrates electronic health record data, trial data, genomics for research purposes. We built it first here within the Harvard community. It's now available at over 200 institutions worldwide. And then the Shared Health Research Information Network, Shrine, connects different I2B2 systems to large federated networks. The I2B2 software can run both on what's called an I2B2 common data model, as well. A lot of institutions are running these on OMA data models as well. The largest network we have is something called ENACT. We'll be talking about that more in one of the later sessions. It connects the I2B2 and OMOP systems across CTSA institutions across the United States with data on over 100 million patients, and it was a model for how we were going to design the Mass APR network. I mentioned the network is live. It has data on over 7 million patients, 10 years of data, and billions of data points. There's a governance framework, which is really core to making this happen. A network oversight committee, which includes leadership from the different hospitals that are participating in this, a steering committee led by Mass CPR and involving lots of others, and then a technical committee with myself and others in this room that manage the underlying technology framework. This is what the user interface looks like. It uses the I2B2 web front end. On the left-hand side, there's a hierarchical listing of medical concepts. These are standard terminologies, about 1 million different things in here with demographics, diagnoses, laboratory tests, a lot of different items investigators or public health users can pick from. You drag and drop them into these query buckets on the right. Everything within one of the query groups is ORed together, and things between groups are ANDed together. It allows you to piece together Boolean queries that can represent inclusion and exclusion criteria of the patients you're looking for. These criteria then get distributed out to the institutions. It takes a few seconds for each site to process it, and then you get a result page back that has the counts from each site and then various kinds of breakdowns. This is some fake data I'm showing you over here. The real system, you'll get counts that are in the millions when you're querying across their whole data warehouses. So here's an age distribution of these patients. We have things that look like NIH enrollment tables for further demographic breakdowns. These are the top comorbidity categories in the cohort that we selected, so we can quickly see what are the other conditions that the patients have. We added some new things, particularly for the public health use cases for Mass CPR, various spatial temporal visualizations. Here we're looking at where patients are from across 3-digit zip code categories in the New England area. You can drill down into Boston and see zip code level data. The underlying data are limited data because it includes these information like zip codes about individual patients, but the software de-identifies it by only presenting it in an obfuscated aggregate way. So we're able to display the information like this to investigators without requiring them to have IRB protocols. Here's a pathogen timeline. So it takes the cohort you selected and it finds for each month various infectious disease rates in that population. And then what's really unique about this network that we haven't done before in I2B2 Shrine networks is pulling in public health information. So here we're seeing in the blue, orange, and green lines, these are rates coming from the electronic health record systems of infectious disease. And the black curve are the actual COVID counts from the wastewater data that's collected from the Massachusetts Water Resource Authority. So we're able to pull in both the local internal hospital data as well as public health agency data and overlay these so you can see the correlations and the time offsets between these two different data sources. So this is kind of the first step we're taking into this, the public health. There's many types of public health data that we can integrate. The wastewater data was already public, so it was less sort of concerning of the potential linkage risks of pulling that into this. There's a nice example of the power of this network to really pull together information from many different hospitals, public health agencies, and bring it all together into a single visualization. The network is brand new. We're just starting to use it. There's already discussion about a lot of the next steps, and I kind of bucket them into technical things, which I find are easier. They may be very technically difficult, but I know we can do it. And then there's harder things where we're dealing with privacy, regulatory concerns, and we may not be able to do them even if there's a technical solution. So on the technical side, there's curating and bringing additional data elements. Things like ICD-9 codes or ICD-10 codes are easy because everyone uses them. Vaccines are recorded in different ways across several different hospitals, and we have to do manual work on that. So as you get into certain data elements, it requires more and more data harmonization and standardization. Another thing is additional reports, analysis tools. I showed you some of them. Bill has shown other kinds of analyses that you can do. So what are the priorities that are being requested by the users of the network? Data update frequency. If you can get data out of the hospitals within a 24-hour period, it opens up many different public health applications. If your data are a month old or a year old, it starts limiting what you can use for it. So trying to get the data as fast as possible out of these systems is really important. And then, of course, bring in other institutions. Some of the harder things are linking records across institutions. So we can do privacy-preserving record linkage to match up patients. That adds to risk. We can pull in biospecimen information. We have to deal with consents and regulatory issues around that. And then ultimately, we'd like to be able to request patient-level data, merge these into enclaves, link the records, and do more complex machine learning analyses out of that. But these are going to require new regulatory frameworks that the institutions allow the data sharing to occur on. And I'll stop there. Thank you.